Okay, so welcome to this section of video 3 of course G04 on mutation detection, where we're looking at structural variation and indel detection approaches using sequence reads. So what we're thinking about here is typically we're dealing with paired end reads. So in that case, we'll have a forward read of a particular length, maybe 100 base pairs, maybe 300 base pairs now, depending on our technology. And we're assuming that they're short reads typically generated with Illumina, and we'd have a reverse pair to that forward read on the same fragment but some distance away. So there may be some sort of insert size of a particular length, perhaps, for example, 500 base pairs, which denotes the distance between the end of the forward strand and the start of the reverse strand. And that might also be the same length as the forward strand that you would typically expect. However, then during QC, we may trim the forward and reverse reads, resulting in a smaller total fragment size. What is worth remembering then is when we try to map these reads to the reference genomes or contigs that we're looking at, the forward and reverse reads will be known, whereas the insert size in the middle will not be known. And we have particular information about the forward and reverse reads, including things like the read orientation. So the orientation, is it looking forward and reverse as it is there? That's the read length itself, the length that's mapped. The insert size when it's mapped, it's an S. Insert size when it's mapped. And then the coverage of reads. So how many reads do we have? Is that expected compared to adjacent regions or the chromosomal or genome-wide average? We also have whether it's mapping as a pair or not, and sometimes it won't. So we have the classic low complexity regions, so repeats of some length of homopolymers, dinucleotide, trinucleotide, or more complex repeats of a particular length. And as you can guess, if these are large, they're more difficult to resolve. So the key thing for simple structure variants, or indeed complex ones, is that if our reads have a particular length, like the ones we had before, and our repeat size is smaller than the read length, that will mean resolving this repeat will be more easy. It won't be perfect, especially with homopolymers, because we know that the polymerase tend to stutter, and therefore estimating the exact size of the homopolymer is not straightforward. It can get more easy as the repeat length has a more precise composition. And then if we're dealing with a repeat where its length is longer than a single read, that means it's more difficult to resolve because we're looking at multiple reads to get a consensus of that information. And if indeed we're looking at a structural variant whose total length exceeds that of the vast majority of our reads there, then it is more tricky because we'll have patterns. We'll have to use the information across multiple reads to kind of get an estimate of what the structural variant might look like. We can't resolve it fully. So that the exact nature of that structural variant will not be known, and we're dealing with many situations where structural variants will be complex. So they might have an insertion and a translocation or some sort of multiple structural variant, resulting in a more complex pattern. So let's look at some of these examples. So here we're looking at the gray read in the middle here. So you got this gray read here, and you can see there it has a homopolymer with many A's before we get this TGC sequence. You can see here that's missing an A here, perhaps in that read that starting A was missed, whereas the bottom one has an extra A, which can happen quite easily with reads. And that would create an insertion in our lower read and a deletion in our upper read. And this is common in 454 and nanopore and PacBio libraries, but less common in PCR-free Illumina ones. We have other examples here of homopolymer again, where we have additional C's that might be inserted, and that might be at the start or the end, depending on the alignment format. So these kind of errors are common in Illumina reads, especially at CG homopolymers. We can look further then at beyond homopolymers at other types of structural variants. So an example that would be a deletion. So deletion means that if our chromosomal sample contained 3 genes, A, B, C, like that, and it was part of a longer region. If we deletion, we might, for example, have the same setup there, except we have A and C only, and we've lost B. B is missing there. Another example would be that we might have A, B, C there. So you have A, B, we might have some insertion inside E, alphabetical length, and then we might have C. So that might be in our sample, and therefore we're extracting reads from this sample and mapping them to a reference of ABC. And this will create differences. Another example is an inversion. So here, there's many types of inversions, but a simple one might be that the region is the same or nearly the same at nucleotide level, but is the other way around. So those are types of inversions, and we look at how we can extract reads from these hypothetical samples and map them back to a reference and deduce the effects that might happen. So if we have no structural variance, the reads map directly to our reference sequence. So we can imagine that we have a bundle of reads from our sample here, reflecting the composition of our sample. And what we're going to do is map them to some reference here. And if they're the same, then they map very well, bar some minor snips. But if we have a different situation where a sample has an insertion that's absent in a reference, that's an additional sequence. So if you imagine here, we have our reference down here, and let's say it's A, B, C, like we had before, and we have the same sample. So we don't know the actual DNA sequence of the sample. But let's just say, for example, this is A, B, oh, we don't really need that, E, and C. So we know that if we have reads coming from here, that we're dealing with paired end reads. So I should actually make them longer. That's the wrong way around. Forward and reverse here. Forward and reverse reads all the way through, even if my diagram is a bit poor. We're imagining that we're pulling reads from this region where each of these regions is longer than a single fragment. So if we map from this region here, if that's adjacent to an A and so on, we'll get lots of reads mapping there, right across A and into B very, very well. But we'll have issues here at this boundary, and we know that they'll map well across C and adjacent to here. What we'll notice is that we'll have reads that are aligned with C, whether you've got the forward at E and the reverse at C. So we get reads will map without a pair here at the edge of C, and they'll map into E. Well, they'll have no partner here because E is not present. E is absent, so I'll have single reads there up here, and the same at B. B into E. So we've got a forward read reverse here and a reverse read in E. So we'll have the forward reads that are unpaired here on either side of the boundary. And depending on our mapping approach, that will also be revealed as a gap. So we'll have limited coverage then at this exact point. So if we plot the coverage here, we'll get, and this is the average here across the genome, we'll find that it's more or less okay as we go through across A with some noise B, that at boundary it'll drop and then it'll go back to the average. So we'll get that gap. And we see that in the read pilot, we'll have a gap there at that boundary where E actually exists in a sample, but it's not present. And we'll also, those are the major ways of determining it. And what we do in practice is we would try to, de novo assemble the unmapped reads, assuming that they're bacterial. So other types, if we have, it's just another diagram, a deletion. So we're dealing with deletion in our sample. So our sample, we got our hypothetical reads here in our sample. And we could imagine that, let's say our sample just contains the A and C. Sorry, my computer paused. A and C here. And then we have our reference genome. So reference genome down here is the same as before. We have A, B, and C. We take these reads, and remember these reads span A into C like that. So those are two forward and reverse reads mapping over that region. We have no A into B, no B into C. So then we map these reads down here. They'll map rel across A, and they'll map rel across C, but then we'll have a gap. We have no B here, and we'll have lots of unpaired reads here at the edges. So if we plot the coverage here, again, if we take the average that we should have, expect to have, what we'll have is some noise here for A, and then a drop, and nothing at B, and then an increase again. So we'll know that this coverage is shows a deletion because there's a gap. And we'll find then what's worth noting is that we'll find that there are reads here that are mapping from A to C. So they'll have a very long insert size going all the way over here, over this boundary. So that will be indication that there's B is absent in our sample. So that's from the insert size length will be dramatically increased. So it looks like that. Another way of viewing this, is that if we have no structure variation, we just got our reads here mapping to the reference there, straightforward. If we get a deletion, then the reads that's absent in our sample, maybe at this point, like there, like that. That'll mean the reads are if this is a read pair, they're much farther away and have a longer insert size because of this gap. And we'll see then that we have this deletion where we have very little mapped. So that's the pattern that we would get. So let's go further and think about indels more generally. So what we would have then if we have a deletion in our, insertion in our sample. So this is an insertion in our sample. The read pair here, if there's a read pair that span the insertion, which is different from the example we gave previously, it will have a much smaller insert size in our reference when mapped. And in contrast, if we have a deletion in our sample, a read pair here, the insert size will be much, much longer in this example. So the insert size can tell us a lot. We will tend to find more insertions than deletions when looking at mutation detection due to this. The penalty of having a key gene lost obviously can be very deleterious, whereas the advantage of having lost a gene may be present, but it will typically have a small effect size. So we certainly will tend to see for viral genomes a strong selective pressure to maintain minimalist genome specific to the biology of the infecting virus particle. Whereas for host genomes, we have less selection pressure, so we'd expect to see a lot more insertions and fewer deletions. So another example is an inversion. So in this case, we can imagine that in our sample, if we have an inversion, so let's take our sample we have here, and we're going to go A, C, B like that. I'm going to extract reads from here. So remember, they're the same for A all the way through. Then we have A into C and C into B. So imagine what happens to the reads when we do that. So we have our reference down here. A, B, C as before. So we can map reads across A, and that's fine. So that region up to there is fine. What we would bear in mind is that we have a gap here at the boundary between this region X and into C, because there's no X into C here. So we've got a gap in coverage at this point. And then if you take those reads that map from A, so we've got a forward from A, that will map okay. And then it will align to a region here at C. So it will have a very, very long insert size. And that'll happen for many reads mapping from A to C. So we'll see that difference. Secondly, if we're mapping from, well, C is inverted. If we note also, that this is an inversion. So this is 3' to 5'. What I should say is that will actually be the other way around. So the orientation will also be changed. If we map from this, we'll note this read is going from the 5' of A into the middle of B, but B in the other orientation. So this reverse read is going to be pointed in that direction as well. And the other one at the 5' end of C, so that's the 5' end of C here. That one will be in the different orientation as well. So also see reads that have an odd orientation. If we think about the coverage across here, what we're going to expect is that there's going to be a gap in coverage here at this boundary, and then also from A into, we expect reads that map from A into B, but there's going to be none there. So if we look at the average coverage across here, what we're going to see is it's okay for A, but then it will drop down here at the gap for B, again that, and then it should be even enough for B into C, and then we'll see a gap at the end again at the boundary of C down here. And we note the reads that map from B into C will be will map okay. They'll just be the other way around here when they're mapping to B, C in the other orientation. So you kind of see that here. So we kind of get the ordering between read pairs can be changed depending on the insert size. So in this case, we have the reference has these different color regions: dark gray, light, medium gray, light gray. And we see the reads that are associated with these mapped to the reference genome, and we've got a sample genome has this inverted region. So if you take this, these two read pairs and map them up here, these ones will, this read corresponds to which one? This one, and this gray read will be the one over here. So we'll see a longer insert size when we map to the reference, and vice versa. This gray read, this black read will be up here, and this gray one will be over there. So it's inverted the other way around. So that will mean that it's not in the correct orientation. And indeed, this black read actually should be the other way around. So they get a change in the insert size. Insert size, the coverage will drop at the boundaries of the structural variant, and then the read orientation is changed. So that's worth noting for inversions. That's unique to inversions that the read orientation changes to. And then we have more complex variants. We got things like a tandem duplication. So we got two A's here. So that means we get 50% of our, well, no, this is our reference. So we get, basically, we'll get 200% of the expected coverage at this region A, because we've got two copies from which reads are drawn for this tandem duplication. We may have dispersed duplications as well from our sample, which then creates issues with the reads that are mapping from, we expect A into B, but not B into A, and then not A into C. So the B into A ones will not map here because that's the 3' end of B mapping here, and that's into the 5' end of A. So that will be a peculiar pattern. And then we'll have unpaired reads, or we'll paired reads that map from the end of A all the way to the start of C. So again, that would indicate a dispersed duplication. And then we may have multiple copies. So that would mean that the easiest way is that the level of coverage here increases at the single copy of A. And we expect, because there are reads here that map from A into A for each of these copies, is that that coverage, if we draw it across here, for our region A, will be at the normal level, and then it'll increase and then back down, but it'll do that before the boundary. So that boundary will be denoted again from this drop back to coverage. So if we have a duplication in the reference, which can happen as well, reference is just an example genome representative we hope, of the larger population, but that's not necessarily the case. We'll find that the coverage is lower. So if we imagine a situation where our reference contained an extra copy of a region which was absent in our samples, like AA, B here, we map it from our sample, which just contains A and B, we'll find that the reads map here well at B into A, they're all mapping well, and then there's a gap here because there's no reads mapping from A into A, and we'll get some of the mapping here. So we'll find that that coverage here is fine all the way here. We'll get a drop, and then it'll, fine, it'll be 100% here at B, 50% at A, we'll get a gap, and then 50% at A as well. So there's other types of tandem duplications. So we can look at things like the local similarity and the changing coverage as mentioned before. If you've got an interspersed duplication, that's more complex in our sample. So we can check the read boundaries again here. That will be detected as a change in coverage at the adjacent part here in our reference, and with an increase in coverage that we would get. And then there's more other variants that are more complex. So we might have a tandem repeat that contains that has many copies called X here, and it may even contain allelic variants like W here, which means that we will resolve well the regions adjacent to this, shown by ABX, and the ones at the end XDE, and we will resolve any allelic variants as well. But resolving the exact copy number of this region can be estimated from the coverage, but it's not necessarily straightforward. So if X's length is greater than the read length, the reads will likely map ambiguously here, which will result in accuracies. So resolving, say, W and other allelic variants within this region will be more challenging. So this is these are universal features that are important, not so much for detecting structural variants, which can be tricky, but also for estimating, inferring information from mapped reads, whether for genomic studies or transcriptomic studies. It has major impacts on our estimation of the true expression rate of a gene. So some ways around this that certain packages use are things like split read mapping. So that means where the middle of a read may be split if it aligns well at two different breakpoints. So an example of it here is if we have a deletion in our absent in our sample, and our reads are mapping fine in the sample because this is a single segment without this block in the middle. When we map into the reference, well, if we have split read mapping, we can map across this deletion area and identify its exact boundaries more easily. An example of where we can use this kind of approach, or the general approach of looking at structural variants, is for looking at trans expression QTLs, quantitative trait loci, whose signals may be associated with alignment errors. And that can happen because we might have some gene A and a gene B, and we might see co-expression because of the sequence similarity of gene A and gene B, because they're very, very similar, and therefore we might mistaken reads from A for those from B. And that might be associated with, caused by reads from A incorrectly mapping to reads from B. And you can see that here we got reads correctly mapped here in black at gene A, reads from A that actually are mapping from B are shown by the red ones. So they're kind of transferred down, and that mistaken approach might link A and B together in terms of their expression when it's actually their sequence similarity that's causing this effect. How to get around this then? So we can look at the coding regions and UTR regions. Here on what we have are uniquely mapped k-mers and ambiguously mapped k-mers. So we can identify the ones that are ambiguously mapped to identify regions that are similar to other regions. So we're looking here at the ambiguous k-mers in detail. And if we look at the ambiguous k-mers with mismatches, we can identify ones that are associated with different regions. So what we can do is count the number of k-mers from gene A, whose alignments start within exons or UTRs of other genes. So these cross-mapping k-mers are useful in that regard. I can estimate the cross-mappability of different regions then across our genome to estimate this more accurately. So these kind of looking at structural variants is really trying to accurately assign reads to regions and resolve what information might be present given the type of reference that is available. And the reference may be limiting or incorrect and may differ. So this is kind of a nuanced area, as you can see. So next we'll talk about the practical overview and implementation.